So today I want to talk to you a little bit about enzymes and what environmental factors can speed them up or slow them down. So the factors that we're going to talk about to start off with are temperature and pH and salt concentration. So let's say the enzyme works really well at pH. Let's do the temperature first. Let's say the enzyme works really well at body temperature, 37 degrees Celsius. Let's say it gets too hot. There might be a hydrogen bond between this amino acid and that amino acid, and maybe one between this one and this one. And if it gets too hot, those hydrogen bonds will break, right? They're going to shake around too much, right? It's shake, 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 and come apart. And so what you've got now is an unraveled protein, when before perhaps the active site was here, and now you don't have that because the hydrogen bonds have come apart. So temperature can disrupt the enzyme. So maybe it works best at 37 degrees Celsius, which is body temperature. And when it gets too hot, this is going to be hotter because this is temperature going up. When it gets hot, the enzyme unravels. The hydrogen bonds come apart because of all that extra thermal energy. So when it gets too hot, these enzymes will denature. That just means they unwind. When it's too cold, it's a little bit different. What happens when it's too cold is you just don't have enough energy for the activation energy. So I have a totally different graph to show you. Do you remember this one where we were looking at energy here, as opposed to this one where we're looking at reaction rate? So the one that we were just looking at said the really high reaction rate happens maybe at body temperature for some of our enzymes, and then when it gets too hot, it denatures and the reaction rate goes way down. This graph is totally different. You would get it in a different way. This is just the energy that you find in a certain molecule. So maybe you're starting with glucose, and maybe you're breaking that down to carbon dioxide and water. I guess we'll have to put some oxygen over here. And then this is going to be the progress of the reaction instead of time. So you need a certain amount of energy to break the bonds in glucose and get to the transition state. But when you form the new bonds to make carbon dioxide and water, you release a lot more energy. So you start with glucose and you end with carbon dioxide and water, and you release a whole lot of energy. And so you need a certain amount of energy just to get started, though. That's called activation energy. So that activation energy is supplied by heat. And so if you don't have enough heat, you can't break the bonds that you need to break in order to get here. So if you're too cold, over here is cold, over here is hot. If you're too cold, you might just not have enough activation energy to make the reaction work. So it's not necessarily denaturing here, it's just that you don't have the activation energy. And as you get more and more heat, you have enough energy to make that reaction happen. Okay, so denaturing is what happens when you get too hot. Denaturing can also happen when the pH is too acidic or too basic for that particular enzyme. So this enzyme might function really well at pH 2, and another enzyme functions really well at pH 7, and another one might function really well at pH 9. So it really depends. For each of these, really, each enzyme has its own specific range of temperature, its own specific range of pH, its own specific range of salt or ion concentration in which it works really well. So let's take a look now at the notes. So we're looking today at effects of local conditions on enzyme activity. So temperature, pH, and I should add in salt concentrations are three environmental effects, are three environmental things that can affect an enzyme. This slide is talking about frequency of collisions. As you get hotter, you have more frequent collisions and you're more likely to make a chemical reaction work. So this part is talking about activation energy. So you need a certain amount of activation energy, a certain amount of heat just to get started with any reaction. All right, this is the graph that I was just, or the chart I was just showing you a second ago, 37 degrees Celsius is the optimal temperature for most enzymes that are in humans. And that's because that's our body temperature. In Fahrenheit, that is 98.6 degrees Fahrenheit, right? So enzymes are going to denature. That's what it says here. Let's move me out of the way. Enzymes will denature when it gets too hot. But what's too hot for enzymes in me is not necessarily going to be too hot for an enzyme in a bacterium that's evolved to live in a really hot thermal vent. So those would be thermophilic bacteria. Then over here, you just don't have enough activation energy in order for the enzyme to work. So optimum means best. Best temperature for us is usually 37 degrees Celsius. So when you explain this in an essay for AP or for me, don't say that 37 degrees Celsius is our body temperature, and so therefore our enzymes have to work at 37 degrees Celsius. Put a little bit more nuance in there. Tell me instead that our ancestors who had enzymes that worked at 37 degrees Celsius were able to survive and reproduce. So try always to explain it, try always to explain it in terms of fitness. So your fitness is greater if you have enzymes that function at the body temperature that you function at, right? And so the optimal temperature for the enzyme that we did the other day in lab, the turnip peroxidase, well, turnips are pretty cold water, pretty cold temperature uh, garden, vegetables. And so they don't work at 37 degrees Celsius. That's like pretty hot. They function better in the spring temperatures, right? So they're going to function probably at a wider range, first of all, than us. We're mammals and we keep a pretty narrow range of temperature. But a plant that lives outside, especially in the cool weather, is going to have a wider range and it's going to be shifted toward the colder temperatures. So different enzymes have different optimal temperatures and also different optimal pHs. So here's an example of different temperatures. So here's an enzyme that works in humans, probably because any enzyme in a human that didn't function at our body temperature would not have increased fitness. It would have decreased fitness, and that organism probably wouldn't survive and reproduce. The optimal temperature for a heat-loving bacterium, though, would be a lot hotter. Be careful here because 77 degrees Celsius sounds pretty okay, but remember 100. Remember in the Celsius scale, 100 is actually boiling, right? So 77 must be pretty hot. Okay, this one's talking about pH, and so remember that your enzymes are proteins, right? Almost all of your enzymes are proteins. And so remember, your proteins are made out of amino acids. And so amino acids, nitrogen, carbon, carbon with an O, each amino acid looks like this. And then they're going to be all bonded together. But they also have an R group. The R group is the thing that makes them different. So this R group is positive, and that R group is negative, which holds the protein shape, holds this part of the protein near that part of the protein. This one's negative, that one's positive. These are different R groups that are attracted to each other, and they're holding that shape so that the protein, the polypeptide chain sort of folds over here. And that maintains the active site. Let's say you have way too many hydrogen ions. If that happens, the hydrogen ions are now attracted to these negatively charged R groups. And then that means the negatively charged R groups aren't attracted to these positively charged R groups. And so the whole structure unwinds, the whole structure unfolds. The similar thing happens if you're too basic. If you have too many OH groups, the OHs will be attracted to the positive R groups, and that means the positive R groups aren't attracted to the negative R groups anymore, and the whole thing denatures or unwinds. And now your active site just isn't the right shape anymore. It doesn't work anymore. Okay, this one is showing you optimal pH in the human body for different enzymes. So your stomach actually is really, really acidic, and the enzyme pepsin works where it's really acidic in your stomach. And trypsin works where it's a little bit basic. Remember, 7 is neutral. So even in your body, you have some enzymes that work at a totally different pH than other enzymes. And you don't say it's because it needs to. Say it's because the enzyme, if it functions at the pH where it works, then the organism is more likely to survive and reproduce. Remember, always try to get it, always try to explain it in terms of fitness. So each enzyme has an optimal pH in which it can function. And that pH is different for different organisms.